In this video, we'll talk about how to prepare shapes for the parallel grid system. This is how the parallel grid system works. Say this crescent is your shape. What it does is that it finds the bottommost edge of your shape, and that will be the location of the first parallel grid line. It will also find the leftmost edge of your shape, and it will add 10 pixels to the left of that to define the line of reference. So this is the line from which we measure distance away from this line. So once it does that, what it does is it finds all these intersections between the grid lines and the edge of your object. After that, it calculates the distance of every point to the reference line, and if more than one point is on the same line, Shape Genie will sum those points into one. Thus, in step 4, we have a linearized version of the object in step 3, such that there's only one value at each grid line position. Then in step 5, Shape Genie does a fast Fourier transform to derive a frequency plot that is a multi-dimensional representation of the original shape. And then you have the option of creating a scalar value as an index to represent the original shape. So there are 3 rules I will be telling you about. They are: 1, that rotations matter, so normalize the orientation. 2, make the rules of orientation consistent for all samples. And 3, add spacing markers to capture spatial context. Rule number 1, rotations matter, so normalize the orientation. Take this crescent as an example. Its concavity is pointing to the upper right, but here are 3 different rotations for the parallel grid system. Each of these 3 will give you a different frequency plot, and none of these 3 will give you the same frequency plot as the original shape. Here's another example. This is an open curved shape. This here is a horizontal mirror image of the original shape. This is a vertical mirror image, and this is a tilted version of the original shape. None of these 3 shapes will give you the same frequency plot, and none of these will give you the same as the original image. So this leads to the question, how should you orient your object before applying the parallel grid system? Well, that leads us to rule number 2, that you should make the rules of orientation consistent for all samples. Take this set of objects, for example. These are all variations of the crescent shape, but they are of different height and thickness. So here are 3 of many options for orienting them consistently. At the top, option 1, I oriented such that the line that connects the tips are oriented to be vertical, and concavity always points to the right. Option 2 makes that connecting line of the tips horizontal, but the concavity points down. Option 3 makes that connecting line horizontal, but the concavity is pointing up. So all of these 3 methods are valid, but then that leads to the question, which one should you pick? Well, without any context as to why you should keep the shapes the way they are in the original image, as in just extract them and apply the parallel grid system to them as they are, without that context, then I would say that choice A is the optimal choice, because you align this object such that its longest length is vertical. This means that the parallel grid system, which always measures distance relative to the left side of the image, it will have the broadest coverage of your shape. More coverage means more information is extracted. More information means the higher likelihood of good, clean results. Now, choice B and C are both valid, but without context as to why they should be oriented this way, I would say that they are suboptimal choices. Regardless of your choice, you need to be consistent within an experiment and between experiments. Here is another example. So this is a set of images in which the shape is open, and they are oriented in various ways. For this, I would align the object such that the line that connects the ends is vertical, and the concavity is all facing left. You can make them face right, it doesn't matter. Just pick one and be consistent. The third rule to keep in mind when applying the parallel grid system is that adding spacing markers will capture spatial context. And this is very important for people who are studying spatial information in the life sciences. Let's take the brain, for example. These outlines are the edges of caverns in your brain called the lateral ventricles. There's one on the left and the right. Keep in mind that the patient in this photo is facing you, so your right is the patient's left. Say you wanted to measure the shape of the left ventricles in blue, and you extracted the outline and you applied the parallel grid system as such. Now, say that you were studying two groups you know to be very different. For example, healthy patients versus diseased patients, or a moderately diseased patient, which will die in 5 years, versus a severely affected patient who will die in 6 months. Now, there's a big difference between 5 years to live and 6 months to live. But say you measured this shape in both categories and you found no difference. While you know that these lateral ventricles are severely affected by this disease. Now, it could be that there is no difference in shape between your two groups, so you'll never find it measuring these shapes. Or it could be that you're missing a spatial context. What do I mean by that? So in this case, one possible spatial context is the midline of the brain. So the brain of humans exhibits what's called bilateral symmetry. There's a left and a right side. And the thing about biological systems, such as this organ, in this case, the brain, when they grow and stretch and move around, they do so relative to each other. So by adding this anatomical marker, say this line, I'm adding extra information here highlighted in green. Okay, what that allows me to do is to capture the spatial context of these two caverns relative to the middle of the brain. And what this little line does is it shifts the reference line from which we measure distance to the left. Because remember, the Shape Genie will always find the leftmost edge of your shape, and then it defines the reference lines to be 10 pixels left of that. In this case, this little extra line here shifts the reference line even further back. And this technique can be the difference between finding shape differences in situations where you would expect there to be a difference, even though your first measure showed no difference or a very weak difference. And yet you know that the clinical outcomes of these two groups is very, very different. What this little line does is that it captures global spatial context. And I call this an anatomical marker in this case because it's part of the human anatomy. And the important thing to remember is that you need to add the same amount of extra information to every sample, meaning this anatomical marker needs to be the same length in every patient, and it needs to be at the midline. And here I purposely made it so that it starts, the bottom edge is at the same level or same horizontal line as the bottom edge of this smaller shape. And this little technique has done wonders. Spatial context is a very important concept in the life sciences, but it's easy to forget because we're so used to measuring volume and area and variations of them, such as surface area. The thing about volume and area is that they are agnostic to spatial relationships, especially within a biological system. But spatial information is more than just area, volume, perimeter, so on and so forth. As an example, both of these mugs are identical because I just duplicated the image and I rotated one upside down. If you measure volume, volume tells you that both mugs are the same. But volume cannot tell you which hand is about to be burned with hot water. Spatial information is more than just volume. Let me now go through several examples within biology of spatial context being important. If you look at the structure of glands that grow within the human body, such as your salivary gland that produces saliva, they follow recurring structures. Even when they branch, they have recurring different types of bulbs. And these bulbs grow at recurring angles that can be mathematically measured. When these rules are broken, when one bulb pushes into another one, we call that cancer or disease. So spatial information, quite important and insightful in measuring shapes. Another example is a phenomenon known as crown shyness. So some trees, when they grow next to each other, they avoid crowding each other and they form nice, neat borders. And so the growth pattern of one tree is not fully understood unless you know where its neighbors are positioned. A third example are moss and lichen, and they grow on typically one side of trees. And the reason is because they prefer shade. That is, they're trying to avoid the most direct sunlight. This is spatial information. And this is a common myth that makes people think that moss only grows facing north. But that's not true. They grow on the side that has the least direct sunlight. So if you're ever lost in the forest, do not try to find north by looking at moss on trees. These are just a few examples of spatial context in the life sciences. It's just as important at the tissue, cellular, and subcellular level as it is at the ecosystem level.